Welcome to this explainer. We're jumping straight into the absolute bleeding edge of bottom-up synthetic biology. We're going to look at the continuous assembly of synthetic life and see exactly how inert, completely lifeless chemicals are being literally transformed into functioning, evolving biology.
It's a question that's been hanging over us for centuries, right? What does it actually take to build life from completely non-living matter? We're talking about bridging this massive, seemingly impossible gap between quiet, inert chemistry and that vibrant, active, dividing state that we call biology. It's an incredible challenge. We aren't just sitting around asking what life is anymore. We're trying to figure out how to piece it together, molecule by molecule.
So, how do scientists actually tackle this? Well, there are really two main ways to go about it. The top-down approach is basically taking a highly complex, already living cell and stripping it down. It's like taking a fully built car apart piece by piece just to see what the absolute bare minimum is to keep it driving. But the bottom-up approach, which is what we're geeking out about today, is a whole different ballgame. That's like building that car completely from scratch, using nothing but raw metal and liquid rubber. We're taking non-living molecular components and assembling them into these increasingly complex, cell-like systems. It's completely reductionist, sure, but man, it gives us some incredible insights into the origins of life.
Okay, so to build from scratch, we got to start with a foundational building block, and that's the protocell. Now, obviously, a protocell isn't a true modern biological cell. Think of it more as a simplified biochemical analog, a really crucial stepping stone for us. It captures the core fundamental features of early primitive life. By engineering these precursors, we're basically building a bridge to mimic that crazy transition phase between pure chemical reactions and the actual dawn of biological evolution. We're literally looking at the bare minimum scaffolding of life here.
But right away, we hit a massive hurdle. Even if we manage to build a simple physical compartment, a protocell is absolutely useless without instructions. So how do we write the biological code for these artificial compartments? For decades, we've leaned on a 44-year-old method of chemical DNA synthesis. And hey, it works, but it really struggles when you get into long sequences and those complex, folding secondary structures. If we're going to build highly complex, customized synthetic life, we desperately need a faster, much more accurate way to write that code.
Now this is where things get really cool. Take a look at this engineered TdT enzyme. This represents a huge shift toward enzymatic de novo DNA synthesis. Instead of just relying on harsh chemistry, researchers are turning to engineered enzymes, specifically this evolved version of TdT. You can kind of think of TdT as a biological 3D printer for genetics. It acts like a highly efficient molecular motor, rapidly synthesizing these kilobase-long genetic sequences. It adds building blocks directly, without stalling out or getting all tangled up when the DNA inevitably tries to fold into secondary structures. It is a literal game changer for writing biological code. And the ultimate, holy grail goal for engineering this TdT enzyme comes down to this one number, 100%. We are pushing for near 100% coupling efficiency. Why is that such a big deal? Well, when you're building a chain of DNA that's thousands of letters long, even a 99% success rate means you're introducing dozens of errors into your code. To actually reliably synthesize life, every single DNA building block has to attach perfectly, every single time. That code has to be totally flawless. But here's the catch. A genetic blueprint, I don't care how flawlessly it's synthesized, is completely useless without a factory floor to read it. You can write the most elegant software code in the world, but if you don't have a computer to run it on, it doesn't do a thing. All of this perfectly synthesized DNA desperately needs a physical, dynamic dividing vessel to actually execute those instructions.
So how exactly do we build that factory floor purely out of chemistry? Well, check out these surprisingly simple, totally non-biological raw materials. This is what's required to build our artificial cell membrane. First up, we've got C10 amphiphils. These are basically simple fatty acids that just love to self-assemble. Then we throw in an MSB buffer, and that's just to maintain the exact right environment at a pH of about 4.9. And then, crucially, we have EDC. Now, EDC acts as a sacrificial chemical fuel. It provides that high-energy spark you need to drive structural changes. It's honestly fascinating, maybe even a little counterintuitive, that just throwing these basic, inert chemicals together can trigger something that behaves so much like life.
And as you can see, this brilliantly illustrates our artificial metabolism in action. We have a simple fatty acid vesicle doing something truly remarkable. It's budding off into smaller daughter vesicles. It's replicating. But what's really mind-blowing about this is that there's absolutely no complex biological machinery involved here. No intricate proteins, no cellular division networks like we see in modern biology. This is autonomous division driven entirely by that chemical energy we were just talking about. It's pure compartmentalization and division, perfectly mimicking the most basic properties of early life.
Let's actually break down step by step how this chemically fueled cycle unfolds. So it starts when the EDC fuel activates, smoothly converting that C10 membrane into these temporary, transient oil droplets. Because of this sudden chemical shift, the vesicle bilayer physically swells up, and that swelling triggers a rapid reaction called hydrolysis, which forces the local production of brand new amphiphile molecules right there at the surface. Eventually, all those excess molecules gather up and successfully bud off as brand new daughter vesicles from the parent. Raw chemical energy is literally inducing structural membrane fission. It's this continuous chain reaction showing us exactly how primitive early life might have first divided.
Okay, so we've seen how we can synthesize the DNA, and we've just seen how we can force a simple protocell to divide using nothing but chemistry. But here comes the next massive hurdle. Biology is notoriously messy. It's complex, it's finicky, and it's super unpredictable. So how on earth do you scale up these delicate, microscopic, unpredictable reactions and turn them into a highly precise, reliable, mass-produced engineering discipline?
Well, this is exactly where the messy world of biology collides with the rigorous world of advanced manufacturing. We are finally moving out of that era where researchers are manually moving tiny drops of liquid around with pipettes all day. To build synthetic genomes and scale up these synthetic cells, we are relying heavily on biofoundries. We're talking highly automated robotic lab hardware. Robotics drastically reduce human error in these super repetitive workflows. They bring a level of speed, precision, and accuracy to experiments that would honestly take a human several lifetimes to complete. And it's this exact leap that's going to eventually allow these synthesized cells to be applied to everyday medicine and manufacturing.
And the absolute crucial framework making all this happen is the DBTL cycle. Design, build, test, and learn. This is basically the heartbeat of modern synthetic biology. Inside these biofoundries, automated lab hardware and robotics are handling the physical, building, and testing phases, churning out high-quality, repeatable data. Meanwhile, advanced software suites are stepping in to handle the designing and the learning. It's this beautifully balanced, continuous loop where every single cycle makes the next iteration just a little bit smarter and a whole lot more efficient. Just to give you an idea of the sheer scale and sophistication of these modern biofoundries, consider this idea. Biodesign automation platforms will employ AI to predict optimal pathways for incorporating synthetic sequences into natural genomes. Think about that for a second. Artificial intelligence is stepping in as the ultimate biological architect here. Before a single physical chemical is ever even mixed in a lab, AI and machine learning algorithms are already predicting the outcomes. They're designing the optimal genetic pathways and optimizing the biological constructs. It's essentially taking all the guesswork completely out of biology.
So we've gone from identifying the absolute bare minimum chemical ingredients you need to bud a simple cell membrane, to engineered enzymes that act like biological 3D printers for flawless genetic code, all the way to AI-driven robotic biofoundries that can orchestrate this entire process at an industrial scale. The gap between non-living matter and engineered synthetic life is closing fast. So the real question we're left with isn't whether or not we can build synthetic life. The question is, if we can fully automate the synthesis of life itself, what are we going to build next?